good day. My name is Dr. Christoph Trautwein. I'm group leader for Metabolomics and Systems Medicine at the Department for Preclinical Imaging and Radiopharmacy at the University Hospital of Tübingen in Germany. We have plenty of projects running human samples from different diseases. So our main research focus is in the realms of oncology and neurology, but also inflammatory diseases. And hereby we investigate, for example, Alzheimer's disease, Parkinson's disease. We are working with breast cancer patients, with hepatocellular carcinoma patients. So it's really a broad range of different diseases where we receive samples from our collaboration partners at the University Hospital. And hereby we apply the IVDR methods. Yeah, well, first of all, it was the wish to contribute something to the research about this really fatal coronavirus disease. I mean, when the pandemic started, a lot of groups also at the research campus in Tübingen started to do their projects. So we had a small project with our partners from the medical genetics where we investigated on the genetic and in the inflammatory scale, we investigated immune cells from COVID patients that were separated from the blood samples. And we really found interesting stuff that metabolites are highly involved in activating these immune cells. This has been published previously. And from the same patients, we also have blood aliquots investigated by IVDR methods. So it's really an additional data set that we compare different layers of omics techniques together with the quantitative IVDR methods from Bruker. And in that field, I mean, we quickly also found out, even though there are a lot of COVID patients, usually it's hard to get patients that are willing to donate their blood or their urine for clinical trials. Because if you're very sick, you have other things to do to care about research. So even in Tübingen, it was somehow hard to get like hundreds of samples from COVID patients. And as I mentioned before, it's really essential for a good statistical power to have like more than thousands of samples, especially if you want to investigate a highly complicated disease as COVID is. So the idea of the network was really fascinating, that we can pool samples from Australia together with samples from Bilbao, together with samples from Northern Germany and the UK and from Italy. And hereby really together, obviously, with the metadata from the patients like age, gender, and so on, we really can better understand what COVID is doing with the patients in the acute phase, but also what COVID is doing on the long term in the so-called long COVID syndrome. About the long-term effects, we have just started. Actually, last week, we started together with our collaboration partners from Heidelberg, a big cohort with almost 600 serum samples from patients that also developed the long COVID syndrome. And we have not investigated the details yet, since the measurements are running. However, what we know and what also other groups reported is that that from long COVID patients, there can be specific organ affections, so that more the neuronal tissue is affected or that more the liver tissue is affected. And metabolites that are classically produced in the tissues, we could identify in samples like urine or also in blood. So right now it's ongoing work, but we have the samples, we have the metadata, and we expect that we will find out very interesting stuff. We just had another clinical project with the pediatry in Tübingen, which dealt about an inborn arrow disease that causes brain degeneration. And in these patients, we found a specific brain metabolite that was excreted in the urine of these patients. So I could really imagine that for the COVID patients that have a strong symptomatic neuronal phase, that we also could see this metabolite eventually in the urine. So it's like a degradation product that it's found there. What we also want to do is together with our partners from the radiology, so as actually I'm working in an imaging department, is to correlate IVDR data from urine and blood samples with imaging data from patients that went to see the doctor because they have a strong like headache or memory problems, and patients that underwent MR spectroscopy and MR imaging of their brain. So in Tübingen, we want to focus a bit to investigate the neuronal effects of the long COVID and to check how the IVDR tools for urine and blood from Bruker biospin could be used for that. So because I mean, if you have like neural problems, then you can go to have an MR imaging maybe once or maybe even twice, but as this is very expensive, you cannot do this every week. So NMR-based metabolomics or spectroscopy, it's straightforward. It's not so expensive if you have the hardware installed, and you can measure in principle a single sample each day. And this is really offers unique possibilities to screen the patient during the disease and also to check how a potential treatment could help the patient and change the metabolomic signature. I'm personally not, however, a collaboration partner, the Medical Genetics, they are exactly investigating this. We might receive some samples of them in the future, but that's not clear yet. I mean, first of all, again, it's like the overall statistical power and also the diversity of a complex disease like SARS-CoV-2. Then obviously it's also a joint venture of unique expertise around the whole world. So I mean, every metabolomics lab has somehow its kind of specialty. So in Tübingen, we work a lot with oncology and neurology. So other labs might work more with metabolic diseases like diabetes. And since COVID has shown to have so many phases that can affect also so many manifestations and phenotypes, it's obviously very helpful to have experts from all over the world that feed their data with their expertise. And this is something I really enjoyed during the network calls of the last month, that we had a very broad knowledge on different fields that we exchanged in these meetings. And this is how science should work. That not a scientist works on his own, but together with other people. And a prerequisite for that, in my opinion, is that data can be compared. And here again, the IVDR methods by Bruker give this opportunity because they're all based on the same hardware and software and SOPs, obviously.